Okay, so maybe I invite Mr. Bilal Ahmed But to give the opening remarks first, please.
Yeah. So if all our participants are there, then I may start with the permission of all of you all are my seniors in that context, very accomplished scientists on both sides. So let me take the opportunity to start this event. Bismillahir-Rahmanir-Rahim. Assalamu alaikum. Good afternoon, ladies and gentlemen. At the onset, I would extend my gratitude to Dr. Lam, Director of State Key Laboratory of Agrobiotechnology, Chinese University of Hong Kong, for arranging this webinar on very important subject of agriculture environment interaction and its implications. I'm also thankful to Dr. Muhammad Anjum, Director General Agriculture Extension Punjab, and our worthy speakers from MNS University of Agriculture, Multan, Ayub Agriculture Research Institute, Faisalabad, PMAS Arid Agriculture University, Rawalpindi, and University of Agriculture, Faisalabad. Dr. Lam, these are very renowned universities working in the field of agriculture, and we are really very honored and privileged that speakers from all these universities are here. These are very huge universities working exclusively in the field of agriculture. Let me admit that I have always learned from Dr. Anjum and other worthy academicians about the issues confronted in by the agriculture sector in Pakistan, and during the public policy, whenever I needed to learn and to take measures that could be taken to address those challenges, I learned from our worthy colleagues all the time. Pakistan is a country with a population of 220 million people with an area of about 800,000 square kilometers. However, less than 30% of land is being cultivated. Though Pakistan is fourth largest producer of cotton, eighth largest producer of wheat, fifth largest producer of sugarcane, 13th largest producer of rice, and 25th largest producer of maize. But unfortunately, productivity of Pakistan is much less than desired. Productivity of all of our major crops is in the middle of the continuum of major producer in the world. Whereas I have found that mainland China is placed normally at the top or at the second position in terms of productivity globally. Of course, there are several reasons that can be attributed towards the low productivity, like poor quality seed, lack of modern irrigation systems, lack of knowledge about balanced use of fertilizers and pesticides, and absence of culture of corporate farming in Pakistan. But in order to feed such a large population with a small land area, there is a dire need to enhance the productivity of crops in Pakistan. Fortunately, we have, as I mentioned, that fortunately we have large body of scholars having expertise in various fields of agriculture. This is a plus point which can be leveraged. However, through collaboration between Pakistani scientists and our friends from mainland China and Hong Kong, this the body of knowledge which our scientists possess, that can be leveraged tremendously. We can learn from the experiences and expertise, the way the Chinese friends have achieved the level of productivity. We really would like to learn from Dr. Lam, your team, and your institution. For my friends from China, Chinese University of Hong Kong ranks among the top 50 university of the world. It was 39th in QS ranking recently. Asim, it was 39th in, yeah. And it has produced several Nobel laureates. You might be surprised to learn that there are several Nobel laureates from this prestigious university in the various fields of learning. The excellence of research and development of Chinese University of Hong Kong has been acknowledged globally and termed as its hallmark. So this webinar basically has been arranged to initiate a process of dialogue for mutual understanding and learning, and to explore further areas of mutual cooperation. I am confident that tremendous potential for cooperation exists between the two sides. I am also confident that this interaction will open doors for further cooperation and collaboration in various fields of agriculture. I would congratulate Professor Lam, Director, State Key Laboratory of Agrobiotechnology, Dr. Muhammad Anjam, Director General Agriculture Research, and Mr. Asim Malik, Consulate General of Pakistan, for organizing this much needed event. However, it may just be the beginning of the cooperation between CUHK and Pakistani counterparts. I would really be looking forward to participate and arrange such kind of joint event in future to deepen our friendship, cooperation, and to enhance mutual learning. So thank you very much. Over to you, sir, Dr. Lam.
Thank you. Talking on the pollution from the ocean to the soil to agricultural field. I'm pretty sure you will have lots of common interests with the previous speaker. So and then let's move on to the next speaker. Professor Dr. Sahir Ahmed is the director of the Office of Research, Innovation and Commercialization and the professor of Institute Soil and Environmental Sciences at the University of Agriculture, Thessaloniki. His research interests include microbial biosynthesis of plant growth regulating substances and plant micro interactions for improving crop yields. Let's invite Professor Sahir to share with us on soil management for changing climate. Please.
Thank you very much. First of all, I would like to appreciate and commend the Consulate General of Pakistan for cooperating and bringing the two labs and the agriculture universities in the country together, and also Dr. Anjam Ali, DG Extension, who coordinated in an excellent way. And today we are together to share our views. So I'm going to discuss soil management for changing climate. Before me, Dr. Ghulam Murtaza, he did discuss different soil issues, which have made my job comparatively easier. So these are the different functions that soil performs. It, of course, delivers ecosystem services that enable life on Earth. Due to the Green Revolution, agricultural productivity increased at the cost of many benefits and costs. And soil quality is actually reducing due to agricultural intensification, urbanization, unstable, unsustainable agricultural practices, jeopardizing our ability to maintain production and food for the future. We have about 60 years of topsoil left. Soil loss is up to 40 times the rate of formation. Dr. Ghulam Murtaza was giving the data from Pakistan. 30% less food will be produced against projected demand. For too long, the world has taken soils for granted, whereas soil is a natural resource that is not easily renewed. These are the impacts of climate change on agriculture. I don't want to go into the detail. Everybody knows what happens due to the climate change. Actually, being soil microbiologist, I'm trying to save time for my last portion of the talk, where I'll be focusing on microbiology. So these are the impacts of climate change on agriculture. These are the impacts of climate change in Pakistan, like increased variability of monsoon, upstream intrusion of saline waters in the Indus Delta, and increased floods and droughts, food insecurity, reduced agricultural productivity, reduction in capacity of natural reservoirs, more rapid recession of Hindu-Kush Himalayan glaciers, and risk to mangroves and coral reefs. These are the strategies to manage soils in agriculture, in climate change phenomena. But my focus today is on microbiology, how microbiology can be helpful for that. So we can use that for abiotic stress tolerance. So one of the important phenomena that is taking place due to the climate change is the abiotic or biotic stresses. Those may be the stresses due to temperature, due to salinity, due to water deficit or excess, or due to nutrients. So abiotic stresses can be tolerated or can be controlled through microbial applications. Detoxification of heavy metals can be done through different approaches. Bioremediation can be a very good approach. And this is the particular area that is the biofertilizers and biopesticides, where we are working on. I will be discussing this in detail, and nutrient acquisition. So this is just to give you an idea. I have selected this picture just to give you the idea that how the microorganisms, they can be helpful through certain indirect or direct mechanisms for improving nutrition, through lowering of ethylene, or through the growth stimulation and plant protection, through enhanced resistance against diseases, decreased sodium uptake, reduced osmotic stress, reduced reactive oxygen species. These are the different mechanisms through which the microorganisms, they can enhance the plant growth. Just you can see, this is the soil environment, and these are different stresses which may be present: pathogen, flooding, temperature, heavy metals, drought, salinity. So if we inoculate with certain plant growth promoting rhizobacteria, they can reduce the stress induced impact. And due to the negative impact of these stresses, you can see the inhibited growth. But these microorganisms, through these mechanisms, they remove the stress effect or the deleterious effect of these stresses, and plant growth is increased. This is actually the area where we are focusing, and I believe listening to your talk, very first talk, even your interest on the soybean and many other crops, this is actually the crux of our work. We are the pioneers in Pakistan. We work on such microorganisms which have the ability of producing a specific enzyme, which is called as ACC deaminase. You being the scientists know very well that this ACC deaminase is actually the precursor of ethylene. And ethylene is a plant hormone. And whenever there is any stress, ethylene production in the plant is increased. And as a result, the root growth is inhibited. So what we do is we isolate such microorganisms which have the ability of producing ACC deaminase. these ACC deaminase, actually this ACC, say for example, this is the PGPR that we have inoculated on the seed or the root. This ACC, due to concentration gradient, goes out of the root, and there this ACC in the rhizosphere changes to ammonia and alpha ketobutyrate, and that can be used by the microorganisms or the plants even. And as a result, the ethylene production in the plant is decreased. And when that ethylene production is decreased, as a result, the inhibitory effect of any stress on the crop plants is decreased. So that stress we have tested, it is very effective in case of nutritional stress. It is very effective in case of drought stress. It is very effective in case of salinity stress. And it is extremely effective in case of temperature stress. So we have such isolates which, when used for inoculating the seeds, they can improve the crop growth through eliminating the stress-induced ethylene production. So that can then result in mitigating, or you can say in coping with the stresses which are induced due to climate change. So this is using that approach. What we do actually here in at our university is that we isolate such microorganisms which have this ability. We screen them in our lab, then we test them under exinic conditions, test them under pot and field conditions, and finally standardize the studies. And ultimately, we have developed these 3 products. This is a bioherbicide. This is another story. Bioherbicide, and you would be interesting. Again, we are the pioneers in Pakistan. Dr. Anjum, it would be of interest for you that we have isolated such strains which are very effective for suppressing the weeds, but simultaneously improve the crop growth. This product we have developed for wheat crop, and we have tested it to be effective against 3 weeds, that is little seed canary grass, then wild oat, and broadleaf dock. Against these 3 weeds, it was very effective, and simultaneously it improved the wheat crop. And this is what I was telling, particularly when I was mentioning rhizobium. This is our innovation, that we have combined the rhizobium with such bacteria which have the ability of ACC deaminase. So ultimately, this improves the nodulation in the leguminous plants. You are working on soybean. So in case of soybean, we have tried to make a combination of bradyrhizobium with such PGPR and an auxin precursor L-tryptophan, and it gave marvelous results as far as the nodulation is concerned and as far as the crop growth is concerned. And this one, the Rhizogold Plus, this has a consortium of such free-living bacteria which have ACC deaminase ability, production ability in them. And this is very effective under stress conditions. So whenever there is salt stress, there will be more ethylene production. Then these bacteria will be effective over there, and they will be reducing that stress-induced ethylene, and as a result, the crop growth will increase. So these are these 2 products where we are using the consortium. These are basically focused on ACC deaminase. And this one, the bioherbicide, in this case, we select such bacteria which these days are called as allelopathic bacteria, which have the ability of hydrogen cyanide production. Then they have the ability of you can say in case of E. coli, they have the anti-metaboloid assay, and they have the lettuce seedling assay that is used for screening these. And ultimately, we have tested it for wheat, and these days we are working for using it for rice. And in addition to that, we are also working on a very interesting approach where we are using cow dung along with sulfur and sulfur oxidizing bacteria. And as a result, what we get is a compost which is of very low pH. And when that product is added, a sort of compost of low pH, when that is added to the soils which have high pH in our country, then it gives a sort of quick shock to the soil, and the nutrients are made available. And to further make it more effective, what we have done is we have one of the issue in our country is the dead poultry. We have big control sheds, so there are many dead poultry birds. So what we have done is we have dissolved those dead poultry birds into a specific chemical, potassium hydroxide. As a result, the product was of very high pH. We mixed that very high pH product with that low pH product. And as a result, we got a very nutritive fertilizer, which is particularly very important. Dr. Anjum Saab, particularly it is very important considering the current fertilizer crisis in our country. And even if we can combine this product with rock phosphate, this can be a very good source to supplement the phosphatic fertilizers. Because your lab is focusing on biotechnology, so as Director Auric, I would say that we do have one Center for Advanced Studies in Agriculture and Food Security. They are working on different aspects of gene editing. Then we have a Center for Agricultural Biochemistry and Biotechnology. So a lot of biotechnology work is going on at campus. So we will always like to have a more exhaustive and you can say detailed discussion with your lab to collaborate on any of the areas of the biotechnology, or I personally would like to collaborate on the microbial approaches for combating the impacts of climate change. And finally, I would also like to share that we do also have a Confucius Institute here at our campus. And I am heading that Confucius Institute here at our campus, which is, the Chinese Language and Culture Center. Thank you very much. And we will be discussing in the discussion session in detail.
Thank you very much. So I'm very much interested in the development of biofertilizers, your microbes, and particularly you're interested in Confucius. So we have a lot of common things to talk about.